Hello again, I'm Ushma Neal from the Journal of Clinical Investigation, and today I'm delighted to be joined by Professor Steve O'Reilly from the Departments of Medicine and Clinical Biochemistry at the University of Cambridge. O'Reilly's research has led to the understanding of the genetic causes of human obesity and insulin resistance. Using modern biochemical approaches and classical clinical observation in humans with profound metabolic disorders, O'Reilly has shown that a person's appetite and feeding behavior can be linked to specific genes. His work has challenged long-held dogmas and led to many new treatment avenues. Perhaps my own ideas about weight gain and my own body can be challenged by our conversation today. But first, let's start with you and your history. Can you tell me a little bit about where you grew up and your parents?
Sure. Ireland, unlike most of Western Europe, didn't benefit from the post-war boom. So Ireland was a pretty grey place for the first 20 or 30 years after the war, and my generation was really the first to go to university in any substantial numbers, and that was true for me and my cousins. My father was for many years an apprentice in a pharmaceutical shop, and when he met my mother, she injected some ambition into him and made him do night classes, and he did that, and he became a trained pharmacist, and then he had to find a premises, and he found it in a blue-collar suburb in the north of Dublin, and that's where me and my sister grew up, effectively in this pleasant but as I say, blue-collar, very ordinary area of North Dublin, and I went to school there.
And how was your interest in science and chemistry, if I'm not mistaken?
Well, I did well in chemistry eventually, but I suppose I was a bright kid. I mean, I read from a very early age, and even in the little comprehensive school I went to, there was some fluster about the fact that I could read so early, and they sent some inspectors in and tried to work out, was there something odd about this kid? And I read avidly and I read widely, and some of the things I read were science related, and I suppose some of the memories of childhood were reading things like asking my dad to buy me the occasional copy of Scientific American and being really thrilled by some of the biology, particularly in Scientific American. And a book that had a huge influence on me was getting Lewis Thomas' Lives of a Cell, which is an American oncologist who's head of Sloan Kettering, wrote a popular book on cell molecular biology. And I think I read that when I was 14 or 15. And I thought it was absolutely fascinating that you could see these big problems, macroscopic problems you could see in life, and then actually they could be drilled down to right down to the molecular and atomic level, and I thought that was absolutely fascinating.
And off you went for a degree in medicine.
I decided to do medicine. I always wanted to, as well as being interested in science, I had a sort of humanitarian desire to do something, to sort of slightly do-goodish view, and I thought medicine was a perfect combination of combining an interest in science with something that would be useful to people and would actually, use my people skills, and indeed almost the sort of, having read a lot in literature, I've always felt that I learned more about dealing with patients through having read Chekhov than I ever learned in medical school. I mean, you can learn an awful lot from the great masters about the subtleties of human interaction, and so I thought medicine's great because it involves a degree of science and a degree of humanities, if you like. And I still believe that to be true. I think it's a wonderful profession.
So you are still a practicing physician, but then what stoked your interest in research?
So I had no idea about research, and Dublin was a wonderful place to train as a clinician, but there were very few role models as researchers, certainly clinical researchers. So I went to England. I was always a bit of an Anglophile. My father had been an Anglophile, I think largely based on the great comedy on BBC television and his obsession with soccer, and he sort of transmitted this English-looking view, which is not that common in Ireland, as you can imagine. There's often been a little bit of friction between the countries, but I grew up in quite an Anglophile household, so I looked for my postgraduate training to go to the UK. And
you were at Hammersmith.
I was at Hammersmith and Barts, got great residency training, and then I thought, for purely careerist reasons, I thought, I better do some research, because everyone has to do research to get some papers on your CV and all that. And I met this wonderful man, Robert Turner from Oxford, who offered me just a year. He had a year of soft money at the time, because I had no research background. I wouldn't have been able to get funds to do a PhD. And Robert gave me a year. He said, I thought you seemed smart and you might do something. And I loved it. I remember that. I almost remember the day, about 6 months in, sitting at a lab meeting, looking out over the green swards of one of the Oxford colleges as we sat there over a long lunch talking about science, thinking, these people are being paid to do this. And I thought, this is amazing. Here's a job where you can be stimulated, have fun, do something that could be useful, and it's a job. It's not a hobby. And so I then decided that clinical academia was what I wanted to do.
So the research with Turner was on genetics?
It wasn't initially, it was on the physiology. We published some pretty highly cited and continually highly cited papers at that time. We got interested in if we looked at people who were first degree relatives of patients with type 2 diabetes, could we discern that they had pancreatic beta cell secretory abnormalities at an early stage? The reason Robert got me into this whole area is that I came to him saying, I'm really an endocrinologist, I'm not a diabetologist, I'm not that interested in I love hormones. He said, well, what do you think insulin is? Do you think insulin is a hormone? First hormone to be sequenced, first hormone to be assayed, first hormone. So he convinced me that insulin was a hormone, and that therefore a perfectly respectable thing for an endocrinologist to want to study. And so I studied insulin. And the work went very well, and we discovered really quite interesting early abnormalities of pancreatic beta cell function. But then the world changed, and the world changed through the revolution in molecular genetics being applied to medicine. And at the epicenter of that was David Weatherall's unit in Oxford, where the first gene linkages to polycystic kidney disease were being found. All this the excitement of this was palpable in Oxford at the time. And so I turned to Robert and said, if I'm going to be serious about it being a clinical academic, I really have to get into this stuff. So can I please leave the lab and go up the hill and spend a year or two or more up learning this molecular biology lark. And Robert was generous enough to and prescient enough to allow me to do that. And so I started, and I ended up doing the first linkage studies in type 2. Very naive, looking back now, underpowered grossly and totally naive and based on southern blotting. But nonetheless, the first applications of that sort of technology to diabetes.
Right. Then from there, you landed at Harvard for a little while.
I did. I had to get the BTA, the Beam to America.
Now, did that experience have any sort of an impact on your approach or not?
It had a huge impact. I really do recommend for anyone working in one country to have a couple of years' experience of another system. Just gives you whatever you do there, because actually, if I look back, I discovered almost nothing in the 2 years that I was in Harvard. But I discovered approaches and ways of thinking, and I also discovered what I couldn't do. And looking back now, I had a great idea of what I wanted to do, and I went to the Mass General, working in a wonderful scientist, Joel Habner's lab, to clone the GLP-1 receptor, which turned 20 years later into the biggest therapeutic target in type 2 diabetes. So I was absolutely right to want to do that at that stage. But I discovered when I got there that I couldn't do it, and partly couldn't do it because I didn't have literally, you need a set, at that stage, you needed the manual dexterity. It was highly technically demanding. And my skills were really to spot interesting patient phenotypes and I'm not a fundamental biologist. I'm much more in the interface between clinical observation and the application of these technologies. And I felt after 6 months of blood, sweat and tears trying to do cesium columns of RNA and failing and breaking things, that I better go back towards my roots. And I went and joined Jeff Lyer in a much more clinically oriented, albeit molecular biological project on patients with severe insulin resistance. And I knew then that that's an area that I could get into. I knew that in Cambridge, which I hadn't been in before, my predecessor, as it turns out now, Nick Hales, had a set of patients with very high insulin levels that they didn't understand the basis of. And I thought that I could come back bringing the tools that I'd learned in Harvard, set up my own little lab, and it was a little lab, me and one technician when we started in '91, and started working on this group of patients to try and see if we could understand why they were so insulin resistant.
So in '91, you did go back to the UK and you landed at Cambridge and you've stayed there since. So what was it like when you were first establishing your lab, and even since, why have you stayed for the bulk of your career?
So Cambridge is a fantastic place, has been for basic science for years, but people don't really realize we haven't really had a medical school until 1975. And when I arrived in 91, it was still an embryonic medical school, tiny. I mean, tiny space. I used to joke that I had to get a chair at Cambridge before I got a desk. I had to work from a lab bench. There was nowhere to do a clinical research facility. You try to bring the patients into your lab, they'd fall over. You try to bleed them, they'd faint. And there were no clinical research facilities. There wasn't, but there were lots of smart people around, and that was, and there were lots of smart basic scientists around that you could feed off. And for me, that was the excitement. I was bringing my clinical questions into a much more basic science environment, and I could get the basic science tools and technologies. In fact, a chap called Ken Siddle, nearly retired now, but Ken was one of the main reasons I went to Cambridge, because he's a world authority on insulin signaling, and I wanted to be able to bring my clinical problems and take a genetic approach, but then work with Ken on the biochemistry, and that worked very well. And a number of us came to Cambridge at the same time. Chris Chatterjee, my longest-standing colleague, really, came to work on the thyroid axis. We're doing very similar. So together, we built up a cohort of young Turks trying to apply molecular biology to clinical problems. And I guess it kind of worked.
And over the years, you've managed to attract any number of trainees and then keep them.
Well, that's unusual. I think that's been a nice feature of the environment. It's a dynamic that's different. I've often thought of it somewhere halfway between an American dynamic and a European dynamic. And in an American dynamic, it's very hard to keep one's trainees, because the American obsession with independence is so profound that that sort of thing is very difficult to create. Whereas in Europe, there is almost the domineering Herr Obergruppen professor can squelch creativity. And what I've hoped to try to do in Cambridge is create an environment where everyone wants to stay, get enough out of staying, feel the synergy brings more than the sum of its parts. And so one of the great pleasures in Cambridge has been to build something from a small number of people into what now is, and we've just got major funding from the Wellcome Trust and MRC to solidify this, but into one of the world's largest institutes for metabolism, diabetes, obesity, which I hope will last after my coronary arteries occlude. So.
That's excellent. One of the things that I've seen in looking over the publications in your CV is that you started and have stayed steady, mostly studying the ideal model organism in the human. Yeah. But over time, you've also started moving towards more animal models. Yeah. Why?
Okay. We are increasingly finding humans with extreme phenotypes where we find what looks like it's got to be a pathogenic mutation. But these are so rare that we don't have enough. An N of 1 that is not transmitted through a family. So it's very hard to prove causality. So at the very least, creating an analogous the identical mutation in a mouse model and showing that there's very clear analogous phenotypes really closes the circle and provides proof that this is the case. And in many cases, this is actually has slightly leaped ahead of the pure knockout. I mean, the one gene I'm talking about at the moment is the P85 alpha, PI3 kinase subunit. We've recently found a human being has a particular mutation in that. It's never been found before. Now, you knock out that gene in a mouse, and the mouse is actually hypoglycemic because the total knockout has massive compensation. The heterozygotes are unaffected. So it was very puzzling in the mouse what the hell was going on. Now, with this human mutation, you make that precise mutation, which you would never have guessed, just a priori make it in a mouse. But by finding it in a human, then feeding it into the mouse, you can make both species in a way inform each other. You can do the right experiments and create the right mutant alleles and really get the most out of that synergy between human work and mouse work. I mean, there are a number of things. I mean, part of the reason for not doing mice is that we had a competitive advantage in the human phenotyping because we concentrated a lot on getting high quality phenotypes, whereas many labs around the world can work with mice very, very well. So why should we try and compete at the, but as we've got larger and as the institute's got larger, it's sometimes you have questions that you can't answer in a human, and you really have to get down to the model organism.
There was one instance where it was the mouse that informed your human work, and that is well, the specific one that I'm talking about is the OBOB mouse.
Absolutely.
With its defect in leptin found or identified by the Friedman lab in 1994. Yeah. And one of the series of studies that your work that brought you the most in the limelight was the study of the Punjabi cousins that were the first humans found with a defect in leptin.
Yeah.
Can you tell us a little bit about that study?
So we've been studying children with severe insulin resistance and getting quite a lot of publicity among pediatricians and geneticists in the UK. And of course, obesity is a cause of insulin resistance. And I'd been alerted to a patient in my own clinic who had severe obesity and very high proinsulin levels. And to cut a long story short, we found the gene in her, a very rare gene. There's only been 5 or 6 people with this found. But that really brought us into the thinking about obesity as a biological phenomenon. But around that time, we got contacted by a clinical geneticist from the UK, around Luton. And that clinical geneticist reported to me 2 first cousins who were extremely obese and came from a consanguineous family of Pakistani origin. And around almost a few weeks before the Friedman paper had appeared in Nature, I thought, we've got to study these kids. And in fact, the human gene sequence had not even been cloned at that stage, but we managed to get skin biopsies from these children and get cDNA out and sequence the cDNA. And as one of these amazing stories, we actually missed the mutation. This is about a year before we reported it, because it was a row of 6 Gs that were very, very densely packed together in those old sequencing gels, and they were so packed together that the homozygous loss of 1 was barely visible. So we went, oh dear, it's not this. So then Sadaf Farooqi, my now professorial colleague and great friend, and Sadaf joined the lab, and the first thing I said to her is, there's some leptin assays available now. Why don't you revisit and just measure leptin in those kids? And she came back and I said, well, it can't have worked. You know, there can't be no leptin. I mean, there's just, she said, well, I think, and so I said, let's get those old gels out of the thing. We got the gels out, looked through, and there it was, having sat there for a year. So then, of course, it became apparent, wow, we've got a potentially treatable form of obesity. This could have a major impact on their health and well-being. And then Sadaf and I went forward and
contacted Amgen.
Contacted Amgen, went ahead and started the trial, and the trial results were dramatic. I mean, it was a miracle. Within a matter of days, these children's food intake behaviors were just totally normalized, and then rapidly. We were very gentle with the first couple of children. They didn't give them a huge amount of leptin. No child had ever been given leptin before, so we had to do it very gingerly. But it became clear it was very safe, and now there's about 20 or so kids, and have all responded wonderfully well, and we're much more aggressive now about treating them, getting them normalized as quickly as possible. So it's a miracle cure, but sadly only for a small number of patients. It's clearly going to have more therapeutic benefit. It's working in lipodystrophy, in patients who have severe metabolic defects because they lack fat tissue and therefore have low leptin too. In those patients, it has a great beneficial effect on their metabolic dysfunction. So it will become a real drug. And then once it's out there, we may find certain subsets. I think there's still life in the old dog yet, in terms of once the history of endocrine treatments tends to be that once a drug is out there, and it then is imaginatively used by interested investigators in a variety of circumstances, other uses start emerging, and other subsets of patients start emerging in which these things are useful.
So have the patients that you started out treating with leptin, have any of them developed leptin resistance?
Amazingly, no. I mean, that is striking. There isn't a single one that has developed, and you imagine even on an immunological basis they might, because they have, some of them, stop mutations that they think and they do develop antibodies to leptin, but biologically they continue to respond, and they have not developed a resurgence of their obesity. Even now, I think the longest time we've been treating the patient is 12 or 13 years.
So have you ever counted how many mutations your lab has discovered?
I think I did the other day. We were trying to work out, discovered or co-discovered, but it comes somewhere between 17 and 20. And when you say discovered mutation, I mean genes in which the first mutation, obviously we found thousands of mutations in particular genes, but I think there were 17 conditions that had not previously been genetically characterized. And that would be either obesity, insulin resistance, lipodystrophy, and some reproductive disorders that we contributed
So a colleague of yours that I was speaking to recently was saying that by the time a patient gets to you, they have been thoroughly examined by their own GP, by specialists, by other geneticists who cannot figure out what the source of their malady is. So how is it that you have had success on at least 17 to 20 different cases? How is it that you're so good at finding these needles in the haystack?
Okay. Well, I think a lot of medicine, of course, is based on assuming that all that can be discovered is what's already known. So a lot of doctors' jobs are to find out, do you fit into an already previously described category? And if the answer is you don't, then they say, well, you don't either. They throw their hands up. And I think that the inclination, I'm not unique in this, there are many good physician scientists now who are both good and able diagnosticians and are able to look at patients and look at their phenotype, if you like, but have enough knowledge of biology to think, hey, if we could really work at things from first principle here, let's try and make some guesses. Now, I had a lot of fun over the years. Making the guesses has been fun. Of course, we're in a totally new era now when you don't actually need to be that smart anymore, because for less than $1,000, you can do a whole exome sequence and give it to a good bioinformatician. And so many of the genes we are finding as a result of a fairly thoughtless process in some ways. Now, there's a lot of thought that needs to go into the bioinformatic analysis and the subsequent linking of that to the phenotype. And I'm not saying, but the world has changed. So I went through a very pleasant decade or more where, based on understanding the biology, I could make some guesses about what might be going on, and sometimes the guesses were right. But now it is a little bit of throw it into the mincer, see what comes out the other end, and actually it's very exciting because we will discover many more things, and we are discovering many more things that way. And of course, what we want to discover is unexpected biology, and in some ways, a criticism of what the approach that we've taken over the years is kind of, we would have known that really, because it's not that unexpected, because that's why you guessed it might have been that, because we knew that this was involved through works in rodents or C. elegans or zebrafish or whatever. So what's particularly exciting is when you can find stuff that you weren't expecting.
Well, the difference is you're the one that actually found it.
I'm not the only one. There are many others, and Rick Lifton in blood pressure, and Helen Hobbs in lipids, and there's a number of us around the world who have. I think the key thing is it's the garbage in, garbage out scenario. And if you select your phenotypes to study to be the most extreme and the most likely to be highly penetrant genetic phenotypes, then you'll find highly penetrant genes. So it's all really about the initial selection of the quality of material, if you like, that you're going to put into your genetic strategy.
One of the main messages that I've seen you give in the media at large is that there's a genetic underpinning to obesity. Do you feel, or have you noticed that over the course of your career, there's been a shift in attitude towards obesity? I mean, why is it that obese individuals are portrayed in the media as being so repugnant?
It's fascinating, and I don't think I'd hoped we may have changed and shifted things, but I don't think we have, frankly. I think that the vast bulk of public literature on obesity is condemnatory and is accusatory, and, as you say, portray the obese as almost uniquely unclean. I mean, there's almost no human condition which carries with it so little sense of sympathy or a sense. It's fascinating, and that has been very difficult to argue against. Even though we've known for 50 years that the heritability is extremely high. I mean, it's really apart from height, it's one of the most heritable phenotypes that we have, and of course it's because everyone has a deep sense of that they understand the nature of their own body weight and why they're lean and why others, and naturally lean people like to think, well, I'm lean because I'm in control and I know how to control myself. So those other people, how could they let themselves get like that? They must have such lack of control, and this is therefore a moral choice that they've made, and it's very easy for it to be confused with moral choice. But it does. It's still. I would like to do a sabbatical, I think, sometime in a philosophy department to try and deal with the nature of the repugnance with which obesity is viewed compared to, I mean, I view it much more repugnant to cheat on your taxes or to cheat on your spouse, for example, or to park, if you're not disabled, parking in disabled spaces. So why are all those things, in a way, much more acceptable than being fat? Because being fat simply doesn't bother anybody. It doesn't. It's just you're there and you're fat. And, why should it cause such a tremendous abreaction? And that's not something molecular biology is going to tell us about. So I think I may need to do a sabbatical in an Institute of Advanced Studies somewhere.
I think you also need a publicist, someone who will help deliver the message more broadly, because it does seem to be pervasive that people assume that with the shift in portion sizes and more processed food and diet and more TV and video games, that has been the sole cause for the increase in obesity.
So you said two very important words there. You said sole and you said cause of the increase. So these are two concepts that people seem to find great difficulty in holding in their brains at the same time. Genetics has nothing to do with the recent increase in the prevalence of obesity. The increase is entirely environmentally driven. And it's hugely important to bring in environmental processes and changes to try and drive down that and reduce. So nothing I say about the heritability says that we shouldn't be doing something about portion size, about advertising, about encouraging environmental spaces that people can be more physically active in. Stop 2-for-1 deals on healthy portion sizes, etc. So all of that is really important, and I'm not on the fundamentalist wing of the reductionist biology argument. What I am saying, though, is that some people are much more susceptible than others, and that in this environment we are, all of the very susceptible are already obese. And we're driving the moderately susceptible into being obese. There will be some that are always resistant. Whether, even in Louisiana, 40% of people are lean.
When you are able to actually discover the underlying reason for their obesity, whether it's a symptom of a greater thing, how do your patients react when you tell them that there's a genetic reason for their weight gain?
It varies. So some are delighted that their understanding that they were intrinsically different has been validated. And some feel that this is now that they have a mark and that nothing that they can do can change that, and therefore they're irrevocably doomed to be like that. So that again is a whole area of psychology and investigation that we really probably should explore in much greater depth, the understanding of how to handle that information to patients about that sort of disorder, because there does seem to be a very broad spread. Sadaf, now, who handles these people on a much more regular basis than I do, would be a better person to ask about the percentage in both. The flavor I get is that more are, if you like, grateful or delighted or pleased to have some even have T-shirts made saying we're the MC4 family from, so they take it very positively and are able to tell people why this has happened, why the family looks like that. So others find it rather depressing information.
Well, one of the colleague who I was speaking to last month about you was saying that he's always taken very careful notes on the way that you deal with your patients and that you treat everyone the same, and including your trainees. So what has been the key towards your approach towards mentoring trainees and obviously being very successful if you've been so magnetic to build your center in Cambridge? That's
a tricky one. I guess I would say the same as I say about the success of genetic studies is about the raw material. One of the things that Cambridge does is it attracts very, very high quality raw material. And I can't make a silk purse out of a sow's ear. But if I have silk purse to start with, if I have the very high quality starting material, I would then work quite hard with them to try to work out what rings their chime, what is it, which bit of the research space are they most likely to thrive in? And is it likely to be in the more translational area, or is it likely to be in the more fundamental lab animal area? And try and give them a bit of time to see what's involved in that.
So a quick last question then. Beyond clinical genetics and perhaps philosophy, as you mentioned earlier, do you have any other vocations that you perhaps would err towards now, or that you would have pursued if not being a medical doctor?
Somebody's asked me that once before, and I remember we had a conversation over a beer one night. So I'm going to answer in the same way as I did then. So my perfect life would have been to have played on the left wing for Manchester United between the ages of 18 and 35. Before then, taking up singer-songwriting a la Tom Waits, or even the best of the Sinatra era, between about 35 and 50. Then, winning my Nobel Prize in Science between 50 and 70, and then finishing by writing the great Irish novel, but a la Gabriel Garcia Marquez, rather than. So I think I would have liked, if you could fit everything into a possible life, the sporting, the musical, the scientific, and the literary would have all featured.
Would have been a charmed life.
Yes.
But you've still led a really excellent one. So thank you very much for taking the time to talk to the JCI.
Okay, thank you.